And then next solution are surrogates. And the classic here is, and part of the reason FDA really has high standards for surrogates is that just because something is correlated with the disease doesn't mean that by changing that surrogate, you're necessarily going to affect the outcome. And CAST study is the classic example. So this is a study where they studied antiarrhythmics in patients who had MIs, because after patients have MI, if you count the number of these things called PVCs, premature ventricular contractions, basically extra beats, it's highly predictive of sudden death, that they're going to have an arrhythmia, they're going to die all of a sudden. So people developed these drugs that prevented that, and they were very effective at preventing PVCs. And somebody had a bright idea, hey, let's do a placebo-controlled study to show that these drugs are saving lives. And there was a huge uproar saying how this is unethical, how can you give people placebo? So after a lot of back and forth, they started a study with the DSMB, and everyone's like, oh man, they're going to stop the study. They're going to start the study, and almost immediately they're going to stop the study. Well, they started the study and they stopped it, but because patients on drugs were dying at a much higher rate. And it goes to show you that surrogates, even though they may be very, very compelling and highly correlated, do not necessarily correlate with clinical outcome. And we're finding that even the well-established surrogate endpoints like blood glucose and blood pressure may not be a great predictor for outcome. And in fact, it may go the other way. So with TNF inhibitors, people noticed TNF levels were elevated in congestive heart failure patients. So they did a study with anti-TNF inhibitors, and it worsened mortality. And when you have a disease and some marker goes up, that can mean that that marker is causing the disease. It may have nothing to do with the disease, it's just an epiphenomena, or it may be body's response to compensate for the disease, and you can't always tell which is which. And in this case, probably TNF inhibitors were compensatory. And then another illusion is people confused by this, although it doesn't affect results of the study or the interpretation, is often people are kind of confused because you do a study and one of the sites shows a reversal. So, the whole study as a whole, there's maybe a 60% benefit, but one site, there's a 60% increased risk. And you say, oh my God, what's going on? How come this happened? Maybe, they were non-compliant with the protocol. But if you have enough sites, the likelihood that you're going to have at least one site that is the opposite is very, very high. With 7 or 8 sites, it's 75% that you'll have a reverse result. So, and same thing, if you do enough studies, even if the drug works, you have to remember it's powered for 80 or 90%. So if you even if you do a study with the drug that works, if you do it enough times, then sooner or later you're gonna get the wrong result. And that's just a function of the power of the study. And then next is, if you look at safety events, because safety events are not predefined in advance, and because people report it in a lot of different ways, it's really important to try to zoom in and out to see if there's a signal. So if you have a disease, for example, that might cause ulcers or arrhythmias, you can't just look at arrhythmias and ulcers because that might not show up. You have to, for example, if you're looking for arrhythmias, you have to look at sudden death, feelings of palpitation, people passing out, because all of these may be signs of arrhythmia. You put it all together, then you may see a signal. Or for ulcers, if you just look for duodenal ulcers, nothing may show up. But if you go to, let's say, severe acute abdominal pain, you may see something. But if you zoom out too much and you say any type of abdominal pain, then you may not see anything. And so if you zoom in too closely, and you look at specifically, stomach at ulcer or nausea or one specific symptom, you may be too close to see it. So it's important when you're looking at safety adverse events to understand that the categories you have to define properly and at the right sort of zoom level. And I think the last one is, if you use subgroup analysis, that can be very, very dangerous. So, for example, let's say you develop a drug for GI symptoms, and you don't see much of a signal, but you think, well, maybe it's because the drug wasn't absorbed. These patients have these GI problems, and maybe it's because the drug never got into the system. So let's look at the plasma levels and look at the subgroups by drug levels. And you do that and you realize, oh, you know what? Patients who absorbed the drug and had therapeutic levels actually benefited quite a bit. Like, oh, that's great. And the patients who did not benefit. So actually the drug works. It's just that it didn't get into the patients. Well, that may be true, but often it turns out to be false because there's confounding. So patients who had milder forms of disease and therefore absorbed the drug well, and they would have absorbed just about any drug well. You could have given them aspirin, they would have absorbed it better. Had higher blood levels, and they also did better because they were less sick to begin with. So you think, oh, these guys did great because they absorbed drug, but it may be true, but unrelated. And you see that like in vaccine studies too. People look at responders and they say, well, only the patients who had best response to Alzheimer's vaccine did the best. Well, Alzheimer's is an inflammatory disease, so you could have given them a TB vaccine and they might have had a better response, but that doesn't mean, you can't tell it's true and related or true and unrelated. So that's one example of the problem with subgroup analysis. Second is, if you only look at patients who completed a study, so let's say you're doing a study on some anti-itching drug and you only look at patients who completed it, then you might get a signal. But the problem is that those patients who dropped out, you don't know why they dropped out. And once again, it could be because they had worse disease, for example, and you're basically biasing the sample. Another way of looking at it is not just looking at patients who completed, but saying, okay, let's just look at the patients who had some effect, placebo and active, and let's just get not just the completers, but do a responder analysis. Patients who responded, and you find that, among the patients who responded, the drug group did, a lot better. But the problem is that there might be dropout issues, differential dropout, or maybe the drug you gave caused adverse events in patients who had worse disease. So whenever you're doing an analysis at the end of the study that's different group of patients than when you started the study, there are dozens of ways where you could have introduced a bias. So it's really important to understand that there's that risk and be very cautious about drawing strong conclusions from those results. It's like trying to figure out, okay, which country is richer, United States or Chile? So you say, boy, we don't have much data, but I've got a friend who works for Mercedes, and he can get us the average household income of people who buy Mercedes. So let's get the average household income of people who buy Mercedes in the United States and average household income of people who buy Mercedes in Chile. When you find all the Chileans who buy Mercedes are millionaires, you say, oh, so people in Chile are much richer than the ones in the United States. That's sort of like a responder analysis. You're kind of selecting a few subgroups that have a particular characteristic, and you may be completely skewing the data. That's why responder analysis and completer analysis are very, very dangerous. And I think I got it in time here.
Thank you, sir.